thank the organizers for having me here and also the speakers for the fascinating talks. What I could add to the discussion, as I thought it was just the emphasis on the viral diversity, the issue that is actually has been alluded to by the previous speakers. As many of you know, the genetic diversity of the has been probably a single most difficult problem for many human pathogens, including the HIV and HCV. For the challenges associated with the genetic diversity of coronavirus, for example, which is, I think, could easily applicable to other pathogens. What is unique about the coronavirus is that we're going to have to look at the genetic diversity at two different levels. The first is the viral diversity in the virus that exists in animals, which it's been described in previous talks, and since the discovery of the first coronavirus back in 1930s, there are many more coronaviruses that have been found in birds, mammals, and humans, which are sort of classified into 4 genera, alpha, beta, gamma, delta virus. And then listed here are the sort of human coronavirus, really coming from the different genera and also the different lineages or clade, if you wish. So based on the current technology, it is actually very difficult, if not impossible, to predict, which virus is going to be the one causing the next pandemic. The second level really has to do with the coronavirus evolution in humans. Although the coronavirus evolution is supposed to be slow because of the proofreading capability of its replication machinery. But nonetheless, we have sort of been witnessing the new variants popping up all over the different countries. I think initially we heard so much early last year about this DG6 or D614G variants. We actually solved the cryo-EM structures of the both versions. The original virus carries the D614 and also the ones actually carrying the G614, of course, the spike protein structure. They actually sort of see the big structural changes caused by only the single residue substitution. And the end of last year, we heard all additional variants, the UK, South Africa, and the Brazil strains, different variants, and we're currently working on the cryo-EM structures of those variants, and hopefully we can actually learn the structure basis for the enhanced infectivity as well as the resistance to the neutralizing antibodies, and therefore reducing the efficacy of the first generation vaccines. So there's really sort of tremendous, and again, at the moment it's hard to predict what kind of, I'm sure there's going to be new variants, what kind of new variants we will be having in the near future. But it was really the tremendous genetic diversity of the viruses is really the big problem, and we really need to be prepared and come up with the innovative strategies to deal with the viral diversity for development of vaccines and therapeutics, as well as the rapid diagnostics.
If I could, thank you very much. Sorry to interrupt you. Thank you very much. As just a brief overview of where you're from, we'll go through the rest of the panel and then we can start our debate. Neon, shall we move on now to Brian Bird? Brian. Now I can't see Brian's camera on. Is Brian here?
I'm here.
There you are, Brian. Thank you. Couple of minutes. We have a slide for you, I think. Thank you.
Oh, yeah, sure. So I don't have any slides because the time was short, but what I wanted to represent in this forum on rapid detection and assessment of novel pathogens is actually upstream of the assays. I want to put that squarely in everyone's mind here. If you look behind me, here's a large collection of straw-colored fruit bats from Tanzania. So I lead projects along with other colleagues at UC Davis and with EcoHealth Alliance across Africa and parts of Asia to do novel virus discovery and disease detection, and particularly focused on bats and rodents as reservoirs of viral hemorrhagic fevers of people. And what I really wanted to stress here is Peter alluded to earlier, you know, our reservoir animals live in very complex communities and structures. And, you know, we as virus ecologists and samplers and detection scientists, we work in a community that we don't really understand, right? The structure of bats and how they live in a cave is very complex. The rodent structures and the way the species move about in the environment are very complicated. And which individuals of these populations come in contact with people, which ones are actually the high risk individuals that we come into contact and things like that. So as we're talking about building new and more sensitive, more complex detection assays, which I think are wonderful, we need to really think about, well, how do we get the specimens into the tubes or onto the sensors? You know, right now in my line of work, you know, we do intense sampling of wildlife. You know, like here, an example of capturing bats in mist nets here in Tanzania. You know, but it's really one specimen, one bat. But that bat is part of a huger community. And I think the promise of these remote sensing platforms and detection technologies will really take us to the next iteration of virus discovery, because, you know, the microbes, the reservoir animals, the evolutionary hosts, they all live in a community, and we need to start sampling the community and move beyond individual animal sampling and detection and testing approaches. You know, here's an example of one of my teams in Sierra Leone doing rodent captures for Lassa fever. And we have very, very in-depth, extensive sampling in that project in communities. But again, we're catching individual animals. I would really like to hear thoughts from the people developing these new technologies and how we can scale this up, really have a network of sensors, a network of pathogen detection devices out there so we can understand not only the viruses we know that are present in the environment, like Lassa fever, Marburg virus, Ebola viruses, but what about all the others, all the other forests of potential pathogens that exist in between the trees of these specific assays? That's where I really would like to see us move towards scalable approaches to surveillance and detection. So, thank you.
Brian, thank you very much indeed for that quick introduction. Let's move on to Catherine, please. Catherine, I can see you have your camera there. We have some slides for if you need them.
Yes. Hello. I don't have any slides for the brief discussion. Again, I'm Catherine at Lincoln Laboratory. Brief intro, we're a Department of Defense Research and Development Center overseen by MIT. I'm technically an MIT employee, and we're charged with developing technologies in support of national security. I want to caveat my opinions today are just mine and do not reflect the laboratory or the DoD necessarily. I've been working on pathogen detection and characterization since the mid-90s, during which I got my PhD in bioengineering with Paul Yeager, developing microfluidic devices for detecting pathogens in low resource settings. And I've continued and expanded this work since joining Lincoln Lab in 2002. So the dear map, I've been working in biodefense and pathogen detection for actually over 24 years. And it's only leveraging that knowledge as well as recent events with COVID-19 pandemic. Here are some quick thoughts I have on gaps or areas of opportunity in the area of rapid and accurate detection and assessment of emerging pathogens. First, and this is staying pretty high level, but I focus operationally, we need a much better understanding at the basic science level of how different viruses and classes of viruses interact with their host at the cell, organ, and organism level, and related, what key signatures of behaviors are really of interest to us functionally. We need this information to develop the right assays to answer the questions we actually want to answer. So if you think about it from a public health and safety perspective, what we really care about isn't, do you have viral RNA in your saliva, or do you have antibodies to a given virus in your blood? But instead, the real question we care about is, Can you infect, could you infect someone with COVID-19 right now? How infectious are you? We use these metrics such as RNA as proxies, but we think we'd want a more relevant proxy directly heading towards the question of active viral load. We don't have these assays, in part because we don't have the foundation of the knowledge to develop these assays and promulgate them in an efficient way. We need a better understanding of the molecular and cellular interactions between active virus and host cells, such as biochemical pathway changes and cell property changes, which we can then exploit to make the assays to ask the questions we actually care about, including looking at how different classes of viruses induce these different changes to allow us to possibly characterize emerging or novel threats. A related question we care about but we can't actually ask is, could you be infected with this pathogen right now? Not have you had the vaccine or do you have antibodies? Those are nice proxies, but we actually care about the functional question. And more research into how we can ask those questions, I think, would be very helpful. Related and building on Dr. Wang's talk from this morning, can we characterize an emerging pathogen based on its impact on its host, whether human or other, as measured with wearable sensors? Are there physiological signatures associated with highly contagious viruses that we can exploit by collection with wearable sensors? And as I wrap up quickly, very different topic, but I wanted to bring this to everyone's attention given the cross-disciplinary nature of this team here. In the world of pathogen surveillance, in my experience, looking at human pathogens, very well resourced, but they're very rare. Plant and agricultural pathogens are much more common actually, and we don't bring the same level of resourcing to bear there. For example, the USDA New Pest Advisory Group typically finds 50 or more new pests a year. They appear all the time. Can we leverage that knowledge to dry run the novel testing and other systems we put in place for human pathogen surveillance? I'm trying to find ways to think about, can we leverage the problems the ag and plant pathogen communities face, right? Bringing resources to bear from the human pathogen world to allow us to beta test and develop better systems and methods and test their efficacy in real world conditions before the next human pathogen emerges.
Catherine.
That's all I have, thank you.
Thank you very much indeed, Catherine. We'll move swiftly on. Stella, have we got Stella here? No, I didn't think, did I see Stella?
I am here.
Yeah, hi.
Good afternoon already, everybody, and thank you very much for having me here. A quick introduction of myself. I have a science technology background. I am now working for a number of years as a director of technology and innovation for Cabrera Corporation. So I represent pretty much the end, the extreme end, extremely important, but extreme end of what you have been speaking about here, right? We have been speaking about how the virus goes from animals to people and the different variants here. And what I represent at Cabrera Corporation is on the building systems, including cruises, including homes, right? Once the virus, unfortunately, is installed in humans, how we can use buildings and building behaviors to detect, contain, and mitigate the virus spread. So I have been working with, I sent a few of you on the science of this, but my specific interest or our specific interest is how to implement the existing and the new research and technology to sense, detect, and mitigate virus situation. From the science, the technology, and also from the emotional perspective. And I'm going to speak a little bit more about them, right? So we all know about buildings. We all may have been tested with temperatures and tested with filters in our homes, HEPA filters, ionizers. All that is excellent technology. It's a mitigation technology already, and all of that consume energy. So we are already putting more emphasis on a problem that is energy consumption that is one of the causes of the problem that we have right now. On the sensing technology that can be applied in buildings, there's a level of maturity that is still very low. Temperature sensor is not good enough to detect and sense when a person is sick and is spreading the virus, but still doesn't have any symptoms. In the building systems, we have the opportunity to detect when a person or where somebody in the community, in the building, has the virus, and on several occasions, right? So we have access control, so access to individuals. We have a lot of the systems in the buildings that we can reuse, for example, in thermostats, in carbon monoxide sensors. There's existing infrastructure, and there's air hunting where all the air passes through, and there's the building management system. So there's the opportunity to sense the virus, if those sensors exist, in several ways in the building systems. Those sensors, a lot of you are already working on those sensors. Those sensors have a lot of scientific breakthroughs, not just from the sensing perspective, but also from the manufacturing perspective, right? So how we make sensors that are reliable, that are accurate, that are specific to certain virus, that will not have variations with temperature, with the level of humidity in a room, with the level of carbon monoxide, and so on. So how we distinguish a good signal from the virus at very low concentration levels to any other signal. So there are a lot of claims of companies that have virus sensors or virus estimations, and in fact, they are humidity sensors. So they detect more humidity variations than virus variations. So that's one of the scientific challenges. How the sampling frequency. Do we access the virus from one person, or we integrate the signals to another person? Another scientific question is the buildings have a lot of different systems. So there are radar technologies, temperature sensing, videos, analytics, microphones, how we integrate all of those signals with sensors that may not be ready or accurate, and how to improve the level of accuracy using the existing system. So those are research questions that we are working on the emotional level, how to manage all of this. And I'm going to stop here.
Beautifully done. Thanks very much indeed, Stella. Thank you. We'll come back to you very shortly when we have our panel discussions. Olgica, now I'm sure I'm pronouncing your name incorrectly. If you're
That's
here,
perfectly fine. It's
jolly good. And I understand you can't put your camera on because of internet at the moment. Is that correct?
I have issues with my internet, but I would love to just say that I am at University of Illinois, and that my research really lies at the intersection of information theory and machine learning, and applications of these two areas to emerging topics in computational biology and bioinformatics. And I don't think it gets any more important than the topic we're studying here. If you can go to the next slide, please. So I was very happy to hear about Peter's questions asked regarding how to determine the number of unknown viruses in a population. I think if all of you just go on Wikipedia and Google a famous problem, which in the machine learning and information theory area goes under the name of unknown species problem, you will know, and those of you that have a background in mathematics and in machine learning, you will know that this has been a focal subject of a lot of research since the 40s in statistics and estimation theory. You probably heard of the experiments where Corbett was trying to catch butterflies in Malaysia in order to estimate the number of unknown number of species, butterfly species. You probably heard of the famous work done by Good and Turing, which tells you how to estimate the distribution, unknown distribution, when you have very small number of samples. And these questions are very statistical in nature, but they're crucial questions we need to answer in this area, because if we can estimate the number of unknown species, then we can figure out how fast or how many more experiments do we need to do, how many more tests do we need to do to come anywhere close to the predicted number of species. Second, since this panel is on testing, you know that it's very crucial to predict which areas of the genetic code of a virus or a bacteria will mutate less significantly than other areas, because you need to do primer design, and the primers should be able to bind to that region and allow you to perform accurate testing. If you cannot predict the regions which will have very low frequency of mutations, then it can become an issue early on in the massive testing efforts that people are trying to use to mitigate the spread of the virus. Next slide, please. Another idea that I would like to put forward, again, from the mathematical and statistics information theory community, is to address questions of scalability. And I think Brian brought up the question of how do we make these tests scalable? And we, again, in our area have known a lot about scaling testing up, because there is the famous work on group testing that dates back to World War II by Dorfman. And the idea is more than straightforward, and it shouldn't be a scary thing to think about. The idea is to group individuals and test mixed samples or mixed saliva, blood samples, and try to determine if at least one individual is infected in the pool or not. And the savings you get in this way are exponential. We are talking about tremendous provable savings in the cost of testing. Second, to answer a question or posed by Brian, I think, you can also use correlations, because you know that some communities, I think Brian brought it up, a large number of communities have highly correlated test results, or are correlated insofar that if one member of the community is ill, of one household, it's very likely that other members are ill. That allows for additional tests, test savings that are allowing you to scale it up further. And this is something that's really receiving a lot of interest in the information theory community, in particular the last year or so. So next slide, please.
Olgica, we'll just have another 30 seconds, is that right?
Okay, sure. So, and what I would like to advertise is also the fact that we would like to have some handle on agent-based modeling of diseases, and how group testing and intervention strategies can help in that way. And that also requires huge involvement from the machine learning community. And I will shamelessly advertise here the work from UIUC, who implemented a massive saliva-based COVID-19 testing protocol called SHIELD. And I'll stop here. Thank you.
Thank you very much indeed. We'll hear from you again shortly when we have our panel going. We have two more panelists we'd like to hear from. David Rizzo. David Rizzo, are you here?
I'm here.
Oh, I can hear your voice and I can see you.
All right.
Lovely.
All right, no slides for me. I'm sort of one of the unique ones in this panel that I actually don't work on human or animal diseases. I work on plant diseases, and it was great to hear Catherine give a shout out to plants in her little introduction there, so I appreciate that. And so when you think of plant health, is really intimately linked to human health through a wide array of different things. We can think of food security, food safety. So we have plant pathogens that reduce yield and reduce food available, human pathogens, toxins that impact humans that are on plants. We have the economic aspects, even on non-staple crops, for example, coffee, that gives a livelihood to millions of people, particularly in low-income countries. We also have the impact of pathogens and other pests on natural ecosystems, which serve as habitats for many of the animals that have been talked about here. So Catherine kind of mentioned using some of the techniques that have been applied to humans to apply them to plants, but I think we can also flip that around and look at things that we've been doing in plant disease research and apply it to human and animal research. And when I looked at that list that Peter put at the end of his talk, almost all of those things apply to plant diseases as well. Particularly things around emergence of plant pathogens. And I think one thing that I've noticed over the years is, one thing that plant pathologists have been doing for a long time, particularly has been looking at the environment. Plants can't get up and move around. They are really impacted by the environment. So that has always been incorporated into studies of the emergence of plant pathogens. Brian mentioned communities. Individual plants in many cases are not worth a lot. A single wheat plant is not worth much. So plant pathologists has always worked at the level of populations and communities, particularly people working with disease ecology and natural ecosystems. And so there's been a lot of work on modeling at many spatial scales, from individual field to global, and incorporating all sorts of things that have been already discussed here, with impacts of climate change, land use change, and then incorporating things such as population genetics of pathogens. There are a number of really important models that have come out, particularly in the last 10, 15 years in the plant disease world. There's been a lot of talk about surveillance, and obviously we do the same thing with plant diseases. Many plant diseases are aerial in nature, and so there have been lots of work, primarily with fungal and bacterial diseases and for aerial pathogens, but taking surveillance in individual fields, scaling those, developing models for how we react to it, how we manage, how we control diseases. And this has been done for a very long time. But we also have the same issues that have been talked about here. We are doing similar surveillance in natural ecosystems and finding thousands and thousands of pathogens, viruses, bacteria, particularly fungi and oomycetes, which make up the most common plant pathogens. But then how do you predict which one of those is going to be the next epidemic? And we have these massive epidemics in natural ecosystems. Chestnut blight, Dutch elm disease, the list goes on and on. Billions and billions of trees have been killed. But how do we decide which one's going to be the next chestnut blight and which one is really not going to do much? So that kind of ties into a lot of things. And then one last one before I get cut off is the other thing as plants as model systems, ethically and expensive. You could do things with plants that you can't do with animals and humans. And I will stop there.
Beautifully done. Thanks very much indeed, David. And we have one more panelist here, Kristian, who I think has only just arrived and probably not even got his coat off yet. So, Kristian, are you here?
I am.
Ah,
yes,
lovely. Thanks for making it. Just a couple of minutes. Just a couple of minutes about yourself, and then we'll get into the debate amongst you all. All right. Thank you.
Sure. Yeah, hi everybody, and thanks for having me on. So I'm Kristian Anderson. I'm a professor at Scripps Research, and like Chris, I lead a couple of research centers funded by the NIH to understand the emergence and evolution and spread of viruses. And I would say my points on sort of research priorities have changed with COVID-19. A lot of our previous research have been very focused on detecting outbreaks, which is obviously critical. But I would say that what I've learned during COVID-19, that even when you can detect it, that doesn't actually mean that you can now handle it. And I think the United States has been an extreme example of that, where in the very beginning of a budding pandemic, we went back to the really sort of basic things about should we even test for this virus, which is absurd to say the least. And I think people like me were really surprised by that. Because there's no question of, you have an emerging outbreak. Should we actually just all go out and get the virus? Do we need to know whether we have the virus or not? Which changed my mind from like, oh, as long as we can detect it, then we're probably going to be good going forward, because then we can try and prevent it. So I will say a lot of my thinking on what is actually important here has changed as a result of COVID-19. I would say one key thing too that has certainly been learning as part of this is to expect the unexpected. Our problem right now is not detection. We can do that. We can diagnose. We can even treat to an extent. We have vaccines. But the problem is that when we have all our pandemic preparedness plan being around flu, was a problem in the beginning, but it continues to be a problem because people are like, well, as soon as we get the vaccine, we're all going to be fine, forgetting that it needs to get into the arms of people, and forgetting that we're actually dealing with a rapidly evolving virus here, which might throw a couple of unexpected things after us. So I would say in terms of research priorities and in terms of how do we actually better deal with these things is, from my perspective, learning to take a more holistic approach where we need to be prepared for the basic things always, which is detection is a key part of that. We have to have things like genomic surveillance so we can actually monitor the virus and pick up these unexpected things that I talked about, as well as having analytics and interpretation tools to actually understand if the virus is changing. Is it an unexpected change? Is it changing in some sort of important way, like we have seen with SARS-CoV-2 and the B.1.1 lineage first detected in the UK, or the B.1.351 first detected in the South Africa, which both are successful lineages, but for different reasons, right? One is more inherently transmissible, the other one is capable of evading some parts of immunity. So I would say from my perspective, all of these things really critical. They're not SARS-CoV-2 specific, right? They can actually be applied to any disease, new, novel, or known already, but it needs to be in place and it needs to be there. So I think those are my comments on that.
Thank you very much indeed. So, ladies and gentlemen, we've met the panel now. The next half an hour or 20 minutes is here for a lively discussion amongst the panelists. We don't want anybody to own any of these questions or spend too long with it. We want to hear a broad spectrum of. So, if you hear your voice speaking for just a little bit too long, maybe you want to hand that across to other people. But I think Nian and Christine are going to sort of moderate that for us. But let's just make sure everybody gets involved of our panel here in a lively discussion. Thank you. So Nian, across to you.
Yes, thank you, Tim. Thank you, all the panelists. So I want to start with the very first question. This question is really for everybody. So I appreciate if you can just respond quickly. The question is, what types of innovative approaches, tools, and devices need to be developed for rapid and accurate detection of emerging pathogens in humans to facilitate pandemic readiness? So
that's a big.
Yeah, will I ask a question to start, please?
Yeah, sorry, that's a big question. Well, I think that I mean, we need to think about it holistically again, right? So we need to have the tools available where the tools are needed. And what I mean by that is that if you look at at the Ebola outbreak in West Africa, for example, there's an example of a known virus popping up in but popping up in an unexpected area. And really importantly, an area of the world where they did not have the diagnostics available to detect it quickly, but also importantly, not a place of the world where you actually expected to find it in the first place. Except though, that when we now look at the ecology and understand where do Ebola outbreaks pop up, we should actually have expected it, right? Because Guinea specifically is one of these places with rainforests as the region, you have non-human primates, like you have the bats, you have all these things that we know actually lead to Ebola outbreak, but we had never seen it before, so we didn't necessarily expect it to be there. So those things need to be there, right? And they need to be where they're actually needed. And then on top of that, you have predictive analytics saying like, well, if you get something that looks like Ebola, you should probably expect it to be Ebola if it's a place like Guinea. So I would say those are one of the key things that you need to have everywhere and need to be available where they're actually needed.
Great, thanks, Kristian. And do you have other panelists who want to share your points, view?
Well, in my presentation, I already did a little bit. Unfortunately, I am speaking and represented when you already have an outbreak, right? So, but the building infrastructures that everybody uses and technology platforms that can correlate data with sensors that may not be 100% accurate, right? So somebody beforehand was speaking about wearables and tattoos, right? How we can correlate that with building systems that may include thermal cameras, radar to detect heart rates that are becoming more available, still science. So that cross correlation of personal and community data can be used to detect when there are variables that are not adjusting, right? So, and it can be used at the personal level to communicate to a person, it can be used at the building level, it can be used at the community level to understand where certain variables may not be as common as possible. So a lot of research, science and technology research needs to happen there.
Thank you, Stella. And we have Abashi, you want to ask you some question there?
Well, I wanted to follow up on something that's been raised by, I think, both Catherine and Stella, but for all of the panelists, we generally think of detection as a binary thing. Is something present? Is it absent? But one of the things that we've been learning, of course, is that that's not true. And so I'm wondering if you could speak to, within your own research, how do we balance this trade-off between speed and accuracy with whatever it is that we're trying to detect?
Great question.
Olgica, maybe I'll have you go first and then we can have others chime in.
Yeah, I think this was probably directed towards the group testing question. I think I agree with Kristian that if we don't know what can be a potentially dangerous strain of a virus or a bacteria, then things are hard, and one should try to do regular testing. We shouldn't wait for an outbreak and for something to become widespread and dangerous to start testing. And I think this is something that the CDC does. It does every year try to predict which viruses will be dangerous and address that issue in the vaccine, the flu shots that you get. And I think from that perspective, statistical data analysis is crucial. And I may be advertising just my field because that's what I'm passionate about. But if you have even small number of samples and you can predict that some viral strain will have a significantly larger probability of being observed in the future than it has, then you know what you should be on the lookout for. And second, scalability, again, you have to have testing paradigms that don't use individual test schemes. And that is crucial, and the mathematics behind it is not scary at all. I think group testing has been tried out in so many areas, including pooling in biology, that it is something that should definitely be discussed with more details in this panel as well. But I don't want to overtake the conversation here. So,
thank you, Chicken. Chris, you want to take over?
Sure, thank you, Jan. I think a few panelists though chimed in. Maybe Bing and Kristian, do you want to top up on what?
There's a few hands raised there as well, I think.
Yes, yeah.
Catherine, yep.
Carry on, please.
Yeah, sorry. So yeah, just to that question of balancing things, I would say the rapid diagnostic testing for SARS-CoV-2, for example, to me is a perfect example of that, because the sort of the normal way of testing this country is diagnostic testing, right? But you actually need, when you're talking about COVID-19 and a pandemic, you need to ask yourself the question, why are you even testing in the first place? And that changes depending on what you're trying to achieve. If you're trying to diagnose patients, well, you need to do a diagnostic test. But if you're trying to prevent somebody from passing on the virus before going into a restaurant, for example, you need to detect something who's infectious. And when we look at a lot of the conversations in the United States around COVID-19 testing, it's because we're so focused on the diagnostic aspect of this. And when we're talking about rapid testing, for example, antigen testing that can be done by the individual themselves, you have a result in 5 minutes, it's cheap and can be done often, is that people have very often compared those to PCR diagnostic and saying, oh, but they're not as sensitive, so they're no good. Which is essentially the same as saying that, if we look at seat belts in cars, I only want seat belts if they can save everybody who gets into a crash, right? The problem there is, again, that the purpose of testing is different, and things like rapid testing, for example, can solve a completely new aspect of testing that you can't do with, for example, PCR-based diagnostics, right? Specifically, do it outside a restaurant before people going in, so the ones that are most infectious can actually be taken out and not go into the restaurants. You don't have to close them, right? So I think this is one key example of where you balance these things, so specificity and sensitivity or accuracy, if you want to look at it like that.
Excellent point. Thank you, Kristian. Over to you, Bing, for just to top up on that same issue.
So I think my general comments in terms of all the rapid diagnostics design is really is from the protein structure point of view. Then one have to be really careful about the protein antigen quality when produced, because remember these are the mainly is using the spike protein in case of SARS-CoV-2, which is one of the viral membrane fusion protein, which undergoes large conformational changes during the viral entry process. So when you produce it, it could actually potentially representing distinct conformational states if you're not careful. So you have to know the structural details about these protein and for your design of the diagnostic test. In addition, with all these variants, I mean, whether the devices developed based on the previous version of virus, let's say original virus, would be applicable to the new variants. That's again a big question. So that's my comment.
Excellent, thank you. I have a question that's come in through CriStorm that that I think is really relevant, and I guess I would love to hear thoughts from everyone, but definitely Brian and Catherine first. And so the question is, of course, around trade-offs and priorities again. And I think, you know, we've obviously got the contemporary challenge of COVID-19, and we've heard a lot about that. But now we're talking about pandemics, so the next likely thing. And we want to make sure, and I think Catherine brought it up in her talk, that there are mutual benefits to populations. So if we look at, for example, the GHSA prioritization exercises that were done to identify the priority zoonotic diseases, a lot of communities were very concerned about novel pathogens in some cases, especially novel influenzas and coronaviruses and arboviruses, as well as viral hemorrhagic fevers. But also there was a lot of mix with older zoonoses, including things like rabies and brucellosis still cause tremendous problems locally in endemic disease. So I think the question is, if we were to redesign a new way to think about this new way going forward with efficiencies and novel technologies, how can we focus on the new while also demonstrating that these other causes are important? Is there a way that these new advances can address both? And I think over to you, Brian, because we've worked together a lot on some approaches in the field that I think seem to do this. So if you can share your thoughts on that.
Sure, thanks, Chris. So yeah, that really cuts to the heart of this, I think, in a lot of ways, where when we're thinking of the next thing, we can get very focused on detection assays and how we would detect that, and sometimes lose sight of the wildlife reservoirs that it's coming from. But I think the driver for all of this is it has to be relevant at the country level, at the Ministry of Health, Ministry of Agriculture and Environment levels in the countries wherever these diseases are emerging. And I think a great leverage point here is to utilize a known classical pathogen of humans to be your lever that allows you to assess the approach, assess the effectiveness of the new remote sensing technology and things, and prove to the governments of those countries that yes, this approach can work. It can have a public health benefit. It could have an agricultural benefit. And then say, okay, well, now we've brought you this technology that can work on rabies, for example. Now let's go find novel filoviruses or novel coronaviruses, ones that we don't know yet are potential pathogenic threats to people. They're there. We need to know the viral or pathogen landscape. So then we can use the predictive algorithms that others are talking about that's much more sophisticated than what I understand. But okay, now we need to be focused on this subset. These are the ones we should think about going forward for even more sensitive and specific assays. So over to Catherine for your thoughts.
Thanks. Just 2 points on that to follow up. I think one approach that has worked well in biodefense is thinking about basically tiered architectures where you look first for a broader semi-specific assay that looks for classes of pathogens or classes of concern, possibly focusing even more on the things about them that raise a concern. And then if you need, bring in more specific assays that take more time. I don't know how relevant that is to this world, but that's certainly something to think about. I want to follow up also on a comment that came up earlier about this idea of binary, yes, no, infectious, not infectious, at risk, not at risk. And we see this rhetoric throughout discussions of the pandemic. And I think it would be wonderful to see a community such as NSF focus on how we communicate risk and risk benefit and trade spaces, because it's completely missing in our public conversations. And we see it everywhere. And I think it's a real challenge in moving forward and living in this new reality where we may have pathogens emerging or re-emerging, or virus vaccines aren't perfect. So I think it's not my area of expertise, but I think engaging conversation at a national, if not international level on what it means to have a risk-benefit discussion would be good.
Great.
Thank you both.
Ian, you have a question?
Right, yes, so just a question.
One last question.
Olgica and for Catherine. So this is a question from online. So this question is specifically about how we can possibly use machine learning and AI to improve the sensitivity and specificity, particularly for different sensor modalities. For
me, machine learning, I have colleagues who are great at that. I'm sure people here are better than I am. Quickly, I would say I know one area is a lack of data. So I think a better categorization of all the viruses out there with some understanding of the degree to which they cause illness in different organisms would help the training. I know that algorithms are limited in utility when they don't have a broad enough data set to really be predictive. So I think a better capturing and integration of our understanding of global pathogens, both human and not, would allow training sets to be more useful, but I'm not an expert there.
Great, thank you, Catherine. And I see a hands up from Kristian.
Can I just ask one more question, just to hear from some plant specialists that we so rarely get some one-on-one time with, David. What can we learn from plants around disease and climate change? Is there work underfoot, and what could we be doing around that?
Certainly, there's lots of work on plant disease and climate change, particularly, again, how it impacts the environment, changes in rainfall, for example. Again, plant pathogens are really impacted by changes in the environment, but also changes in land use and how all of that is going on. So I know there's a lot of modeling, particularly in natural ecosystems, but even in where crops can be grown from that standpoint, and movement of plants to new locations. All of that is being incorporated.
It's a lot to build on, sounds like,
Yes.
which is great. Thank you.
I am very conscious of time. Is there one very important question that needs to be asked? We've got a great panel here, but time is pressing us on.
Yeah, I mean, I think that we have questions that could go on for like a week and a half, I think.
Exactly.
So feel free to cut us off. We want to be mindful.
You're okay. Thank you. Thank you very much indeed, panel, for that. I hope you're going to join it. Keep staying with us for the rest of the day. We're going to go to some breakout sessions in a short while.